Attendees are in listen-only mode.
All right, hello everyone, and welcome to today's webinar, All You Need to Know About Clinical Trials and Cooperative Group Studies. I'm Sharon Worrell with Fight Colorectal Cancer, and before we get started, I want to thank the rest of the Fight CRC team, Michael Sola, Danielle Burgess, and Andrew Wortman for all their help getting the webinar up and underway. We also are, of course, thrilled to have Dr. Sam Lubner from the University of Wisconsin joining us today for the webinar. After the webinar, you can view the recording at fightcrc.org/webinars. And after the webinar, you will get an email with the links to the material and also to a survey. And if you fill out that survey, we will send you an I Heart Booty bracelet. During the webinar, you can ask a question using the panel on the right side of your screen. And finally, follow along with us on Twitter using the hashtag CRCWebinar. Fight Colorectal Cancer has a comprehensive library of resources for patients, survivors, and others touched by colorectal cancer. Your Guide in the Fight was updated in January 2017 and is a detailed 3-part book discussing everything from dealing with a diagnosis to treatment decision-making and more for stage 3 and 4 CRC patients. The late-stage MSS CRC Trial Finder is a one-stop place to find and learn more about high-impact trials for CRC patients. The current data are limited to microsatellite stable, or MSS, and stage 4 CRC patients. The list of trials curated on the Finder are sourced from the clinicaltrials.gov website. To learn more, visit trialfinder.fightcrc.org. And just want to make a quick note that the information and services provided by Fight CRC are for general informational purposes only and are not intended to be a substitute for professional medical advice, diagnoses, or treatment. If you are ill or think you may be ill, see a doctor immediately, and in an emergency, call 911 or go to the nearest emergency room. Fight CRC never recommends or endorses any specific physicians, products, or treatments for any condition. And finally, we're very happy to have Dr. Lubner with us today. He is a GI oncologist at the University of Wisconsin Hospital and Clinics. His research interests include clinical trials in all GI tract cancers, and he is actively involved with clinical trials in esophageal and colon cancer. He is the faculty co-leader of the Internal Medicine Interest Group and serves on the State American College of Physicians Council. Dr. Lubner has given local and national talks both on his research and educational endeavors. So thank you so much for being here. Dr. Lubner, I'm going to hand over the controls to you now.
Okay. Here we go. All right, I hope everybody can hear me okay. I've got show my screen. I think we're rolling. Okay. Thank you, Fight CRC, for all the work you do and for having me join the group today. I wanted to spend a few minutes talking to you about clinical trials and cooperative group studies. In general, sort of their conduct, what they can do for patients, and also describe some of the limitations of clinical trials and why we conduct them in the ways that we do. So the objectives for this talk are really to understand what clinical trials are and understand that a phase 1 trial is different from a phase 2 trial, is different from a phase 3 trial, and understanding the differences between those three, both in their design, their intent, and the goals of each of those phases of trials. Also to understand why cooperative groups exist, what are things like ECOG, SWOG, what do those acronyms mean, and why are they necessary for diseases as common as colorectal cancer. And then we'll take some time to bemoan why it takes so darn much time and money to answer what should be a relatively simple question when looked at from a 10,000-foot view. But of course, as we all know, it is much more complicated than that on a patient-to-patient basis. So why does this stuff matter? It currently costs around $1 billion to successfully bring a compound from basically from the bench to the bedside. Most drugs fail. There are estimates of anywhere between 1 in 5,000 to 1 in 10,000 drugs that are first compounded versus actually brought to market. And it takes about 8 years for a compound to go from human testing, phase 1 testing, to approval. Now, currently, we're in sort of a bonanza phase of clinical trials and drug discovery. So there are some exceptions to that, and we'll talk a little bit about the accelerated approval process and how that might happen. So the key question when it comes to clinical trials and clinical trial design is, how do we decide what works? We think that if we pour money into research and preclinical models, that we can start to find candidate drugs and candidate pathways to figure out how to make a disease better. So we think about in vitro, or cells in a dish, as a start. And before I go much further, Otto von Bismarck is the German fellow with the pointy hat in the corner of the screen. And he had a great comment once upon a time, which was, 2 processes in which you never want to see being made are laws and sausages. So I would include clinical trials with laws and sausages as things that are sort of dirty and things that you don't want to watch how the process is. You just kind of want the end result. And so this process from preclinical data through in vitro testing is messy, and we're going to expose some of that mess for you today. The process of high throughput screening basically takes some of these in vitro findings and tries to translate them to clinical use. Basically, if you have a disease model, you'll throw a ton of compounds at these cells in a dish to try to see if any one of those compounds will make the cells in a dish die. From there, you can take those cells and put them into an animal, sometimes a mouse, sometimes other kinds of mammals, to see if you can actually create what's called an in vivo model. And you give the same compound to the mouse in the hopes of seeing if you can make the tumor shrink. You want to do that and then get a sense of tolerability, because you'll also see what actually happens to the rest of the mouse when you give them a compound that's potentially toxic. There are tons of improvements in this area of what's called translational science right now, which are sometimes we can skip the animal step and really create a 3-dimensional model of a tumor in say a gel or in some other kind of matrix where we don't have to treat animals. And there are cost and ethical considerations that are always in play when doing experiments on animals. So if we can find non-animal models to get access to good science that then translates into humans, boy, we're all the better for it. The hard part is there is tons more work to do, because we can kill cells in a dish way better than we can kill cells in people. And there's a lot more of translational science that needs to continue at the same pace that it currently is. So while we've made a lot of strides in the preclinical modeling era, we still have a lot more to do to get those models better translated to get drugs to patients. So I want to walk through the different phases of clinical trials, and I have 3 kids. This is a picture of them, and they don't learn anything if I just give them facts. They only learn through stories. So I'm going to tell you guys a story of one specific drug and how it went through the process of clinical trials. This may be a drug that some of you have been on, or you've had loved ones who've been on it, because the story does have a happy ending in the sense that the drug did get approval. So this story is ripped from the headlines, and Law and Order is up at the upper right-hand corner, because right around the time that Law and Order was launched in the early 90s, some scientists both in New York and in Israel found that epidermal growth factor was important in tumor growth, both in cells grown in culture but also animal models. And in 1988, it was found that epidermal growth factor receptor could be blocked. So cell surface receptor, something on the surface of cells, would respond to a signal that was circulating in the serum, and you could block that signal with an antibody. And that antibody would cover the binding site of epidermal growth factor receptor and force it to not be bound. And we thought at the time that that would be a mechanism by which cells exposed to a drug would die. The name of that compound was IMC-225. That was basically, the IMC comes from the company that founded it called Imclone. The 225, I'm not entirely sure, but so IMC-225 was found as a candidate drug from preclinical modeling to go into phase 1 clinical trials. A phase 1 clinical trial simply is a dose finding or toxicity finding study, and often is considered a first in human clinical trial. So basically, we take a drug that has been tested in animals and in a dish and thought to be at least biologically plausible. We think that the drug hits the target. Phase 1 trials have very little to do with an assessment of efficacy. They are quite simply an assessment of safety. The idea is we start at what's called the LD10, the dose at which 10% of the animals exposed to it died. The reason why the LD10 is chosen is because if we started all the way at what we thought would be the highest dose and ended up being too toxic to patients, that would both be unethical but also unwise from the standpoint of it would kill the drug in safety analysis very early. So the LD10 is thought to be biologically plausible and potentially effective, but more importantly, we would consider it to be a safe dose at which to start. And so in phase 1 clinical trials, typically the patients who are enrolled in phase 1 trials have refractory cancers, cancers that have already been treated with everything that's on the shelf. But the patients themselves are still feeling okay and are interested in trying something that is not yet on the shelf to give them access. And this really takes a ton of altruism on the part of patients and their families to say, you know what, we don't have a better option. We want to contribute to the science of cancer and cancer treatment. And with open hearts, they will try to get access to these drugs. It is a high reward possibility in the sense that if we find a drug that actually hits, then these patients would get access to it well before anyone could on an open market. So the endpoints of a phase 1 clinical trial primarily are toxicity, and then defined in the maximum tolerated dose. So all the patients who are enrolled on the study get access to the same drug. There's no placebo arm. There's very little randomization on a phase 1 study. And we know what dose and schedule every patient is being treated, largely so we can determine the toxic side effects of the experimental therapy. So what's called a classical 3+3 design, where we basically put 3 patients on a dose level. We see if any one patient had an excessive toxic response, and if they did, then we expand that cohort to see if that was just a random event or if indeed that dose is too toxic. So I'll walk you through this if you can see my pointer. If there are no toxic responses, then the drug gets escalated to the next dose. And then 3 more patients are in at the next dose level. So if there's one toxic response, well, then the dose is expanded to 3 more patients. And if there are no other toxicities at that dose level, well, then they can escalate to the next dose and enter 3 more patients. If there are more than one toxic response at the dose expansion arm, they stop and say, okay, the maximum tolerated dose was the dose below the level where 2 patients of 6 had a serious toxic response. If in a 3-patient cohort, 2 or 3 patients have a toxic response, the trial is stopped, and we consider the maximum tolerated dose to be the dose previous. Now you can imagine there are some doses where the starting dose is too toxic, and that you get sent all the way back to the drawing board to say, okay, we need to figure out why this drug was so toxic, Or did we overshoot on what we thought the LD10 would be in animals and patients? So the key thing is to try to find a dose that's biologically effective, but also tolerated well enough to proceed. There are some other modified designs where we try to get patients escalated to a biologically effective dose early on in the process, so we're not giving patients lower doses of drug than we believe to be effective. So one of the key things about a phase 1 study is trying to escalate the dose past the biologically effective dose while maintaining safety. Success in a phase 1 trial basically is if the dose is tolerable and beyond a threshold where in a preclinical model we think that the drug is biologically effective. So that's a key thing, and we often in a phase 1 study will use what's called biomarker assessment to see if in a pharmacokinetic analysis or a pharmacodynamic analysis, basically did the drug get into the bloodstream, and did the levels in the bloodstream match or exceed the ones we thought would be effective in a dish. So that's that biological plausibility. And then the tolerability is, of course, the safety assessment. And then only in a second question do we ask, did patients respond? And if so, why? Was there some similarity between responders? Either did they all have the same type of cancer? Did they all have colon cancer? Did they all have pancreas cancer? Because remember, this is all comers. This is just refractory solid tumors. Every once in a while, there's a disease-specific phase 1 study. But now, more and more, we're thinking about having biologically similar tumors. As the FIGHT CRC folks have already mentioned, microsatellite stable colon cancer appears to behave differently than microsatellite unstable colon cancer. So what is it about those two groups that's different? And if we find a drug that responds in both, what does that mean? If you look at all comers, there's a historical response rate of about 5%. So 1 in 20 patients on a phase 1 trial on average will have some kind of benefit. Sometimes we have more, sometimes we have less. If the phase 1 study has a targeted both population but also a preclinical model target, for example, KRAS wild type patients in colon cancer, there could be a dose expansion cohort to say, okay, we really want to test how safe this drug is and potentially how effective it is in a dose expansion cohort limited to patients with KRAS wild type colon cancer. Thinking about IMC-225, in the phase 1 study, 2 partial responses were seen out of 13 treated patients. Now remember that IMC-225 was first found in the early 90s. And the data for the phase 1 study was finally published in 2000 in the Journal of Clinical Oncology, which is the big nerd journal for medical oncologists. It's what we read like people would read the New York Times or the Washington Post. This is real news. This is real data. Based on these responses, phase 2 studies were proposed in colorectal cancer and also head and neck cancer, where the same EGFR was thought to be very important in the development of cancer. So a phase 2 study logically follows after a phase 1 study, basically saying we know the dose and the schedule, but now how effective is the drug? So for patients in whom we think that it would make sense for there to be a response, at this safe and tolerated dose, let's give it to them and see how many tumors we can make shrink. So that's the signal of effectiveness that we're looking for that we can try to more broadly apply. So whereas a phase 1 study is a couple dozen patients, phase 2 studies are typically bigger by a factor of 3 or 4, but they're still pretty small. Sometimes as few as 40, as many as 100 patients. They are also open label on a phase 2 study. You got to know what the patient is getting. Every once in a while, they're randomized. Sometimes if you have 2 candidate drugs or a combination therapy that you're proposing, sometimes you can test them both at once to see if the combination is more effective than either drug in isolation. Phase 2 studies are still typically heavily pretreated or a late line population, largely because we don't want to expose patients for whom we already have FDA approved therapies without some significant biologically effective mechanism behind it. So the endpoints of a phase 2 study are typically things like response rate, how many tumors of a percentage shrink, or what's called progression-free survival. Are we, if tumors were supposed to grow in 6 months, but we find out that it's taking them a year or two to grow, that is a significant finding if we're slowing the disease biology. Additionally, what's called the disease control rate is often reported, and the disease control rate is a summation of what's called partial responders, folks whose tumors shrink more than 30%, complete responders, folks whose tumors disappear entirely, or the rate of stable disease, the folks whose tumors stay about the same size. So the disease control rate basically is trying to look at everybody who at least got some kind of disease stabilization from the proposed treatment. So you'll often see those things reported as a response rate or a progression-free survival or PFS. So when you see those, response means shrinkage, progression-free survival just means it didn't grow. So a real home run would be in patients who had been through all previous therapies. A response rate that is higher than 15 or 20% would be considered a real shocker. And it would really try to get folks onto a fast track where we would want to really investigate that drug quickly to try to bring it to market faster and check and see, can we get this out there for patients to get on to see if we can make the disease shrink. So sometimes after a home run like Carlton Fisk is hitting here, we can get FDA approval after a fantastic phase 2 study. Everybody remembers this home run from, I think it was 1976. They don't happen very often. So these kind of home run studies are few and far between. IMC 225 was thought to have one of those home run stories, interestingly, with a response rate of between 9 and 20% for EGFR overexpressing colon cancer. There were articles in Time Magazine, in the New York Times, and 60 Minutes advocating for rapid access to this drug through what's called compassionate use protocols, basically saying this drug is not FDA approved, but we know it works. And so we have to bypass the approval process and get patients access to the drug. And it was thought to get approval on a fast track basis through the mechanisms already in place at the FDA. So there were standing ovations at national meetings. There was all kinds of excitement about this drug in around 2001, 2002. However, FDA approval was held up because the data that was reported from the ImClone company was found to be incomplete. And on external review, a lot of the benefits that were proposed were not realized when reviewed by an independent investigator. And this drug is why Martha Stewart went to jail, because if you guys remember, she owned significant stock in ImClone and basically sold before the report came out from the FDA saying we're not going to approve this drug. So she was guilty of obstruction of justice, among other things, and other people at the ImClone Corporation were actually sent to jail for insider trading. Interestingly, however, this initial lack of approval didn't kill the drug, because it couldn't be denied that in a subset of patients, there were remarkable responses that couldn't be explained. So in those patients where a response was identified, what was unique? What made them different from other patients? And so we went back to the drawing board and had a better understanding of how the disease biology was taking place, how the cells were dividing faster than their surrounding cohorts. And interestingly, on data reanalysis, there was some suggestion that finding a subset of the population with colon cancer that did not harbor a KRAS mutation, boy, we could really find those heavy responders and get them access to this drug. So by better understanding this pathway, so this is the EGFR signaling cascade, and blocking up here is what we thought would stop the process of cell division from happening. The mutation in KRAS just downstream from EGFR, basically think of a mutation in KRAS as a gas pedal being stuck right here, and the signal would be sent to the nucleus to grow and divide independent of anything that happened upstream. So if you blocked up here, the RAS signal would still be sent, whereas in folks with a normal RAS isoform, if you blocked up here, the signal would not be sent downstream. So we thought that that discovery identified a population for whom IMC-225 could be uniquely effective, and equally importantly, folks who harbored a KRAS mutation, or any other RAS mutation for that matter, would not benefit from treatment with IMC-225 or anything like it. So going back to the drawing board, the effectiveness in KRAS wild-type tumors, they went through similar phase 2 and 3 studies and found to be effective. So FDA approval for cetuximab came through a phase 3 study showing a survival benefit in heavily pretreated patients. It took another 3 years to conduct that study, but it was done. So after you have something that gets preliminary FDA approval, you start using it for everything to try to maximize, in many ways, for lack of a better word, the market share of this drug, trying to use it in different settings, using it in post-operative treatment, using it in first-line therapy. So really trying to do all the things to figure out where can we use this drug to get access to that drug for our patients. So phase 3 studies are used after showing safety and efficacy, but they are very costly because it requires a comparison against existing treatment. This is where a placebo-controlled randomized clinical trial takes place, because without randomization and without some kind of control, you don't really know whether or not the drug under investigation has the effects that you think it does. So of course, these studies are very time-consuming and very costly. And rather than a phase 1 study where there's a high risk, high reward for patients, this is a high risk, high reward for companies. So I mentioned a couple slides ago that it took a phase 3 study for cetuximab to get FDA approval. And hundreds of patients were enrolled on that study. The study itself required a ton of regulatory oversight, including data safety monitoring, and really like all clinical trials, but really specifically on a phase 3 clinical trial, the protocol has to be very tightly adhered to ensure that you're really looking specifically at, did the drug have the effect that you thought it did? So we think that phase 3 studies are considered a gold standard to prove efficacy or not of a drug, but of course there are examples where it hasn't worked out the way we thought. Typically, these phase 3 studies are best run by some kind of neutral party, because if a drug company is running a phase 3 clinical trial, they have a direct interest in the success or failure of that trial. However, a phase 3 trial can't be done without a company because we would need access to the medication somehow. So some combination of an independent neutral party funding the data analysis and the company in kind providing drug is what we think a really good phase 3 trial would look like. So there are tons of different ways to design a clinical trial, but this sort of block diagram sort of describes how a biomarker-driven study can best be done. So the key questions of randomization: do we do it based on disease type? Do we do it based on stage, age? What do we do? What is the effect size we're looking for? Are we looking for a response rate that's 20% greater than what's already out there? Are we looking for an improvement in survival that's 6 months longer? Or are we looking for a cure? What does that mean? Is the randomization ethical? Are we somehow discriminating on some way in which we couldn't predict? For example, if randomization was performed by the patient requiring access to broadband internet access, we would be unknowingly discriminating against patients who didn't have broadband internet access. And sometimes those design issues are raised by you guys as patients or as basically community representatives in clinical trial design, patient advocates, to see, hey, is this going to be generalizable to all patients? The question of offering crossover, what if the drug works so darn well that it's unethical to give people a placebo arm anymore? Writing in crossover in the protocol is very important. And then is there unblinding at the conclusion of the study? Will the patients themselves know what they got, and will they know what their results were? I think that's an interesting ethical question. All of that depends very specifically on how the protocol is written. Sometimes unblinding and crossover are allowed, sometimes they're not. So phase 3 studies really is a brass tacks endpoint in cancer. Do patients live longer as a result of this drug or not? And so really survival time, or time from initiation on treatment to death, is the main endpoint. Sometimes it's progression-free survival. We can use that as a surrogate. But when we're talking about a phase 3 or a registration study, the FDA really wants to see that hard endpoint of life or death being on there. There is a significant safety reporting component. That's in phase 3 studies. That's where we get a lot of the things that you hear the talk to your doctor about. And I'll fly this ridiculous picture from Cialis marketing up there. But all of those things that you're asked to talk to your doctor about are toxicities that came up during some form of the clinical trials, and most commonly in a phase 3. So a registration trial is basically the kind of phase 3 trial that the company is going to send to the FDA for approval or not. So I want to give an example of some of the clinical trials where we've used cetuximab even after FDA approval happened. So the Crystal study was a first-line study where we used cetuximab in combination with 5-FU and irinotecan. And it took 1,200 patients to answer the question, and it showed a response rate of 59.3% and an overall survival improvement of about 3 months in KRAS wild-type patients. Interestingly, another first-line study showed a similar improvement in response rate from about 50 to about 60%, but the overall survival wasn't changed. It was thought that it was because it was added to FOLFOX and not FOLFIRI. Around the same time that cetuximab got approval, Avastin got approval a couple of years prior, so it was thought that, hey, look, if one's good, 2 has to be better. Interestingly, those results were worse. Patients had worse progression-free survival, more adverse events, and so that combination of cetuximab and Avastin actually failed. We don't use that combination because of these results. And then an adjuvant study, cetuximab was given to post-operative chemotherapy for patients with stage 3 colon cancer, and it did not improve disease-free survival. It didn't reduce the risk of recurrence more than FOLFOX alone in patients with resected stage 3 colon cancer. So think about that, right? That cost per patient for enrollment on a clinical trial for these drugs that can vary from a couple $100 a dose to tens of thousands of dollars per dose. The cost per patient for all the regulatory oversight, et cetera, is between $25,000 and $40,000. When you look back at the math on these 4 studies, 6,142 patients were treated there. And so that's on average an outlay of anywhere between $153 million and $245 million trying to ask various questions about where are we going to use this drug. And this is just a sample of dozens of other studies used to try to see where we could fit in this drug in the treatment of patients with colon cancer. Around the same time, another drug, panitumumab or Vectibix, was marketed by Amgen, and similarly confusing results were seen, which was to say In some patients with KRAS wild-type tumors, this drug worked great. But in other patients where we thought by all rights it should have worked, it didn't. And why is that? The other question was, why didn't it work as adjuvant therapy? If it works so well in the metastatic setting, why doesn't it work post-operatively? And those questions as yet remain somewhat unanswered. So after all of that stuff, after thousands of patients and billions of dollars, cetuximab did have FDA approval for the treatment of KRAS wild-type metastatic colorectal cancer. And ImClone, as a company, was sold to Lilly for $6.5 billion. They have since marketed it as a good first-line treatment for patients with metastatic KRAS wild-type colorectal cancer, and I use it all the time because it does improve survival. Those findings that were surprising of the adjuvant treatment, those, unfortunately, we still haven't answered. So we have learned that response rates are better with cetuximab, but that overall survival is changed only by a couple of months. We've also learned that there are a ton of mutations in KRAS that mean that the drug won't work. So HRAS, NRAS, and BRAF, those will equally render cetuximab ineffective. And while responses are great, we can make these tumors shrink, they are short-lived. The progression-free survival is somewhere between 6 and 9 months for most of these drugs. So that even if we get a response by about a year's worth of treatment, we're going to be looking for something else. Of course, there are statistical outliers. I have handfuls of patients who have been treated with this medication for a couple of years and have as yet not seen progression. However, the vast majority of folks have progression events in that 6 to 9 month time frame. We also learned that cetuximab and its cousin drug, panitumumab, work about the same, with less rash overall with cetuximab and fewer infusion reactions with panitumumab. All of that is to say that after all of those billions of dollars, we're still stuck with a drug that improves outcomes somewhat, but not the cure. So why does this matter? As interested patients and as family members and as patient advocates, we're all interested in maximizing efforts to improve outcomes for patients on clinical trials. So the process is messy and not always straightforward. But if you're reviewing information that you're reading for yourself or for a loved one, some of the sort of sciencey questions you have to ask yourself are, okay, is this clinical trial asking a valid question, and is the trial designed to answer that question? Now that takes a ton of statistical sophistication to figure that out. A lot of times, one of the key questions that we have to figure out is, are these results reproducible? Can we replicate these results in other patients to make sure that everything's valid? And then when you're looking in a clinical trial capacity, is this study worth the risk? Is this drug potentially effective enough for me to put myself or my loved one in harm's way? Or from a societal perspective, what good is it to get a drug approved if ultimately it's going to be $750,000 per dose for a survival improvement that's a month long? Those are really hard questions, and I think questions that as a society we have to figure out. Because to me, there is no substitute for helping people stay alive longer, right? That's sort of a no-brainer. But then you ask at what cost to the patient? And that's where it really has to be part of shared decision-making to say, hey, patients, what was this drug worth to you? That sort of endpoint-driven stuff where quality of life questionnaires and patient-reported outcomes are becoming more important in phase 3 studies, where it doesn't only matter if patients are living longer. Those quality of life reported outcomes are going to be as important in the next phase of clinical trials that are to come. And I really think advocacy populations like you guys are so important. So thanks for being part of that. So how do we actually do this? It takes hundreds or thousands of patients to answer some of these questions, and we need cooperative groups to figure out how to develop these drugs faster. So cooperative groups are groups of doctors and hospitals willing to work together to answer these important questions, like does drug X improve outcomes in colon cancer? So there are these things called the ECOG Akron Group, Southwest Oncology Group, or SWOG, the Big Ten Cancer Research Consortium, National Cancer Clinical Trials Group, Gynecologic Oncology Group. There are tons of different cooperative groups out there, all really dedicated to trying to get better science access to our patients. So the advantages of cooperative groups are that we can accrue faster. We can get lots more patients exposed to these drugs, and it's not limited to folks who live within 60 miles of the University of Wisconsin Hospital. We think that the results are more generalizable if we can find them in a variety of different practices. If we can get tons of people to agree to practice the same way, well, boy, we can then say these results reflect real life clinical practice. Some institutions are really good at some of the preclinical modeling. Some institutions are really good at radiology. Some groups are really good at surgery, and we can leverage all of the strengths of different hospitals in a cooperative group study to try to improve the care of our patients. Now, disadvantages of cooperative group studies is that it takes doctors a long time to agree to practice the same way. So sometimes the trial design process is very arduous, and it takes weeks or months and many conference calls to figure out, okay, what is the population we're going to put on? What is our randomization strategy? So we can minimize these last three things. So we can minimize the lack of uniformity or different techniques at different places. What we really want to do is have everybody run in the playbook the same way, because it takes standardization to make sure that the results are generalizable. And that takes a lot of staffing to get it right. So we think that ideas from investigators, thought leaders within a cooperative group go to a steering committee, usually sort of a high council on a mountaintop of esteemed investigators and thought leaders saying, yes, this study makes sense to conduct and I want to be a part of it. So the steering committee gives a yes, no as to whether or not it'll proceed in that group. And the idea typically needs a sponsor. The industry will provide the drug, but then in terms of the science, who pays for some of the biomarker analysis? We figured out the importance of microsatellite instability in colon cancer only through biomarker study that was not necessarily funded by the companies that make some of the immunotherapies. Government funding is sort of a gold standard because we believe them to be the least biased source of funding in this situation. So once a go-ahead is received from a cooperative group, whether it's a phase 1, 2, or 3 study, the members of the group decide whether or not to open it at their sites. And while it seems like a no-brainer if it's gotten higher up approval, there is a process that needs to go through. What is what is the enrichment of this particular disease type at our site? Do we have another competing study open at the same time? So those are the kinds of things that come into play as to whether or not any individual member of a cooperative group will open a study at their site. So once the go-ahead is achieved and the accrual is complete, once everybody gets enrolled on the study and everyone conducts it the same way, the findings are presented and published, and our big national meeting is actually this weekend down in Chicago, where we're going to go over the results of clinical trials from the last year that the data is now being presented. So what about this cancer moonshot thing? The goal of the cancer moonshot is to make discoveries faster and to have them happen at a lower cost. So really the Cancer Moonshot Initiative has a lot to do with the development of immunotherapy, which is making a ton of inroads in colon cancer now, and trying to figure out who the populations are that need access to this drug because it will be so effective for them. And is it one of those things that the science is that hard, or is it the infrastructure? So the Cancer Moonshot is also trying to overcome some of the infrastructure pieces that makes running clinical trials somewhat arduous, basically mandating that if a study is approved at one site, you can open it at another site with relatively minimal review and re-editing of how the study will be conducted. So one of the real breakthrough ideas that's happened in the last couple of years is what's called the NCI match trial. And this really is turning some clinical trial design on its ear, saying that rather than treating colon cancer the same, or any cancer the same based on anatomic sites, maybe we should be treating it based on DNA. If we can come up with personalized cancer therapy, boy, we would be better off. So we hope that through high throughput analysis of patient DNA samples, that we can find patients with a specific KRAS or BRAF mutation or a PIK3CA mutation, and find drugs that would target those mutations or those pathways. And by using that sort of broader way of thinking about cancer, that maybe we can begin to make inroads where previously there weren't any. Now, the hard part about that is if we're making common cancers into a series of very uncommon ones, it might make clinical trial conduct harder down the road and clinical practice harder down the road, where physicians and patients will need advanced molecular diagnostics to see what flavor of colon cancer they have. And we will have to slot them in with different treatment based on one mutation versus another. I'll tell you, I think as a system, we stand at the ready to do that stuff fairly easily. We just simply have to know, is it safe and is it effective? And that's why the match trial is going to be so important, where we basically look at a tissue biopsy and try to target the pathway in question. So that's just a brief glimpse into the sausage making that is clinical trials. And so I hope that now you have a sense that our ideas in clinical trials come from good preclinical modeling. Funding can come from a variety of sources. And we really need to increase our throughput, but we can't lose quality. It really is like making a sausage in that regard. And that it's going to take a ton of people invested in one goal to try to fix the problem that is metastatic cancer of any variety, but in my world, colon cancer specifically. I mean, how hard can it be, right? This is a design of basically an integrated circuit of cell growth. And any of these targets in red, if mutated, can promote cell growth. And so we have to look at the whole integrated circuit to see what's going on. So thanks for your attention. I'll spend the rest of the time taking questions.
Thank you so much, Dr. Lubner. Let me just pull up my slide here. That was a great presentation. Thank you so much for taking the time to walk us through clinical trials. Just a reminder to folks who are tuning in, on the bottom right-hand corner of your control panel, there's a box for questions. So if you do have questions, feel free to type those in at any point, and I'm monitoring that now. So we do have a couple questions to get started. The first one is about how long it takes to get onto a clinical trial. So if you find a clinical trial, what kind of process is it, and how long does it typically take until you're actually recruited and on the trial?
That's a great question. Thanks for asking that. So a couple of things. Different protocols will have different language associated with it, where you have to be more than 2 to 4 weeks out from the last line of chemotherapy. So that's sort of a structural delay to make sure that any toxicities that are found are truly due to the study drug and not the previous treatment. But once you sign the consent to be on a clinical trial, so you go see your doctor, they talk to you about a clinical trial, and they'll typically give you a consent document that you can review and sign yourself and say, I want to be part of this. Once that signature is there, then the study staff can really go through your chart in detail and make sure that all of the eligibility criteria are met. For example, some of the novel immunotherapy agents, patients with a history of autoimmune disorders, things like lupus or Crohn's disease, are not eligible for those trials, because if they have an autoimmune disease and we turn their immune system on even higher, they're going to suffer adverse consequences. So that's why that review process takes place. And that typically takes a week or two to get done. And then the process of randomization happens, and that typically takes about a day. So from the signing of informed consent to actually getting your first dose of drug, somewhere between 2 and 4 weeks typically elapses. Many times, the protocol will mandate that patients, once they sign informed consent, have to get their first dose of drug within 4 weeks, or the whole process has to start over again.
Thank you so much. That kind of is a great intro to the next question. We have someone who wrote that they had to wait about 6 weeks for their first appointment. Is there any way to expedite the process?
Yeah, I think a couple of things. First, working through the first treating physician's office. From a clinical trial standpoint, boy, that 6 weeks is lost for you as a patient, and it's lost for the trial as well. So a lot of those structural delays we really want to avoid. So one of the things that I would recommend for patients is to, when you contact the academic center's office, you say, hey, look, I found this trial on clinicaltrials.gov, and I'm interested in getting on it. My last treatment was this date. Can you please expedite a referral for this specific clinical trial? Now that's an extra bit of homework on the patient end, and some of that's a little bit unfair. But boy, our institutional standard is we really try to get people in within a couple of weeks, largely because we know that your time is so valuable. But of course, you know, everybody's at a national meeting this week, so there's not a lot of new patients being seen. Some of those structural delays happen where, you know, people have competing clinical responsibilities, or there's a ton of patients with colon cancer that are running around and there's just not space for them. But I would also argue, if you're looking at a specific trial, you can advocate for yourself by saying, this is the specific trial I want to get on, and I'm willing to sign consent to have my chart reviewed ahead of time to determine if I'm eligible for the trial or not.
Thank you. We have another question that came in here about when do the results of a positive trial become the standard of care, and what is that process and how long does that typically take?
Right, so that process is somewhat variable. But once there's a positive phase 3 result, many times within a couple of months, a positive phase 3 result very typically turns into FDA approval fairly quickly. Time is money on that one from a company perspective, and time is time for a patient. So one of the things that if there is expected to be significant regulatory hurdles to clear, many times drug companies will get what's called an expanded access protocol up and running while they're waiting for FDA approval. Basically to say, hey, we have this positive phase 3 result. We can get patients access to drug and we will provide it. Some of those expanded access protocols are out there. Sometimes they're even referred to as compassionate use protocols. It is kind of like a phase 3 study in the sense that, you know, it is being used in a very specific population. And the company and the investigators will want access to the patient data. How did people do as a result of getting access to this drug in an expanded access way? So sometimes those things are out there, particularly because we acutely feel the frustration with administrative delays while drugs are being shepherded through the approval process. I don't know of any specific expanded access protocols that are out there in colon cancer right now, largely because we're waiting for some phase 3 results to be published in the next few weeks.
Great. Yeah, that'll be exciting to see what gets published coming up here. Kind of going back to those first few questions, are there wait lists that some institutions have in terms of clinical trials that are very exciting for patients? And if there are wait lists, how are those addressed?
Yeah, so especially for a phase 3 study, we try to take all comers. For phase 1 and phase 2, where the endpoints are a little bit more cautious, where you're looking for things like safety and preliminary efficacy, that's where wait lists can come into play. So what'll happen is a patient's eligible, but there won't be an available slot. But then they get put on a wait list, and once somebody either falls out of the study or there is an additional cohort opened up, so they want more patients on the study, then those patients who are on the wait list will be preferentially slotted into those spots. So the process of being on the wait list is exactly the same as being on the clinical trial, just without actually being on the medication. So you basically, you sign consent, you go through the process, they review your eligibility and say, yep, you're eligible, just wait right here for a spot. And sometimes that takes just a couple of weeks. Sometimes that takes a couple of months. It typically doesn't take much longer than that, but those are much more for the early phase clinical trials. Other wait lists for later line trials where people want to get on a study that's not yet open, you kind of have to wait for the study to be open before it's you can even sign the informed consent process. So we won't have wait lists for that kind of thing, largely because I don't feel like it's ethical to have people wait around saying, oh, don't worry, we'll have it open next week. And what if it's not? And then they've wasted a week, especially if they have other treatment options available to them.
Great. Thank you. I think we have time for maybe one or two more questions. So you mentioned, Dr. Lubner, that clinical trials are for patients who have already tried all the current available treatments. Are there trials that are open to patients who haven't used all of these options? And are there trials that focus on things other than treatment? So for example, like along the cancer continuum, quality of life for cancer recurrence, and do those follow the same process?
I'm sorry, I guess I'm not understanding. Give me another snippet of that question.
Let me see. So it's kind of a two-part. So for patients that are on clinical trials for treatments, are all trials only for patients that have already been through multiple different types of treatments? We'll start there.
Right. Yes. So no, there are within any individual protocol, there are you can specify what line of therapy patients are treated on. So sometimes there is a trial that is restricted to patients who have never received chemotherapy before. There could be another trial that's patients who have tried it and their tumor has progressed after one line of treatment or 2 lines of treatment. And then after that, it really becomes sort of refractory solid tumors. So yes, you can really specify what line of treatment it is, because certainly patients are going to be in different places who've never received chemotherapy versus folks who've received 1 line versus 2 lines. So yeah, that's really specific to the individual clinical trial, largely because we want the population to be in about the same place.
And then the second part, are there clinical trials that are geared more towards side effect management, I guess would be another way to phrase that?
That's a great question. Yes, there are. I think two points. First is, within any given clinical trial, there is a lot of flexibility for how specific side effects are managed. And that's based on good clinical practice. That's based on biologic effects of where we think the side effect came from. So, for example, using cetuximab as an example, folks who have been on cetuximab, you guys know that you can get a pretty bad rash pretty quickly. And it took some interface with our colleagues in dermatology to figure out whether it was doxycycline or clindamycin gel or steroids, what the best way to fight that was. And that actually was written into various clinical trial protocols to say, this is how you should manage grade 1 versus grade 2 skin toxicity. Increasingly, it is becoming equally important to design trials specifically looking at side effect management. So saying, okay, we're going to treat this patient with this drug, or this group of patients with this drug, and we're going to manage this group with this anti-nausea medication, for example, and this group with this one, and see which one's more effective. So yeah, we have improved cancer care especially in the supportive care realm by running exactly those kinds of trials.
Great. Well, thank you so much, Dr. Lubner. We are at our hour, so we'll go ahead and wrap up. I just want to thank all the listeners for tuning in. And again, you can watch the recording and listen to it in the webinar archives on fightcrc.org. So thank you again so much for your time and for a great presentation.
All right. Thanks, guys.
Thank you.